Good morning, everyone. I will be discussing about membrane transporter proteins. The name itself gives us hint that these proteins are involved in transport of some molecules. And the term conformational switches indicates that to transport these molecules, these proteins undergo different shapes and conformations. So I will be talking about how blue waters allow us or enable us to study these membrane transporter proteins and then allow us to get insights how the functional mechanism of these transporters works. So our cells are in liquid environment. So the exchange of molecules between these environment is important to be for the normal functions of the proteins. So transporters are the molecules that allow us the exchange of molecules between the outside and the inside of the proteins. And these transporters are present at the plasma membrane or in some other internal organelles. So these transporters are present in both in prokaryotes and eukaryotes. So in humans, approximately 400 membrane transporters has been identified and broadly classified into two classification, ABC transporters and solute carrier transporters, where ABC transporters are the transporters that perform efflux functions of transport mechanism, which means like it involves the it should take out the toxic compounds from the intracellular from the cell, whereas the solute carrier transport, it allows the intake of molecules into the cell. So in order to transport molecules, these transport proteins undergo different conformational changes. So it has to open towards the extracellular region such that it can recognize the substrate molecule, and then it closes at both the ends, and then it opens towards the cell such that it can transport the molecules to the cells. So here I will show a small video where, so this is the substrate molecules or a molecule that has to be transported from the extracellular environment to the inside the cell. So this molecule is recognized by some transporters. And since this molecule is transported against the concentration gradient, it needs some additional energy to transport these molecules to the cell. It gets these additional energy in the form of proton. So binding of proton involves conformational changes such that this molecule is transported to the cell. So today I will be talking about bacterial peptide transporter, which is a close homologue of human peptide transporter that transport dipeptide and tripeptide amino acid molecules that are essential for functions. So this here I am showing the topology of these transporter proteins. It belongs to the MFS, which is major facilitator family of proteins. And then the helices 1 to 6 belongs to the N-terminal domain, and then 7 to 12 belongs to the C-terminal domain, and the N-terminal and the C-terminal domains were connected by two short helices A and B. So how this transporter works? So when it is open to the cell, the substrate molecules and the proton comes and binds to this transporter, and it undergoes conformational changes, and then it finally releases the substrate to the cells. So till date, two structures were available in one conformational state, which is IF is the invert phasing state, and the essential dynamics between the various states are still not known. So here I am showing the 3D structure of the PepTSO, and then here it shows the probe radius, which means probe radius is nothing but the measuring of volume along the pore channel. It shows that it's closed at the top and then open to the cell, and essentially how the transition from one state to other state is not known. So we performed molecular dynamics simulations. So the protein is embedded in the lipid bilayer, and then we neutralize it. And then, so we have performed 5 microseconds of accelerated MD, and then we got different starting structures. And then we performed canonical molecular simulations for 55 microseconds. So this is a huge amount of data. So we can't analyze these data using like so one by one. So we use some package called Markov state model to analyze these large data. So blue waters enables us the simulation of such more long time scales to understand the functional mechanism of these transporter proteins. So here, so the computational time is much effective, so we need to use it in a much effective way. So here we have a protocol called adaptive sampling approach, and then here in the right hand side we have an approach called single long simulations. So consider this as a landscape of a protein, and then we start with multiple structure and perform short simulations and cluster them based on some geometric data, and then again select the less populated structure for the next round and then perform simulations. So by performing this kind of adaptive sampling simulations, we can sample the entire landscape of the protein in a short amount of time compared to running single long simulations. So this is a very long video, so I'll just skip this. So analysis of the data. So we analyze the simulation data using Markov state model. So Markov state model is nothing but a kinetic approach that allows to discretize the high dimensional data to less dimensions and then cluster them into kinetically relevant states. So once we get the states, so now one can construct the transition probability matrix and then find out the number of times we are moving from one particular state to other states, and then find out and one can estimate the probability of transition from one state to other state. From the probability transition matrix, we can find the rate of transition from one state to other state. So using this approach, we analyzed our data. So this is the conformational landscape we got for PepTSO, so which has been obtained by measuring the gating residues. Gating residues are nothing but the residues that allows or stabilizes particular conformations or switching of the conformations. So based on two distances, so here the top distance is measured as in the x-axis and bottom distance is measured in y-axis. Here we have seen like, so increase in distances at the bottom side shows means it's open to the cells. When it is closed at both the sides, we call it this as occluded, and when it is open to the cell extracellular surface, we call it as outward facing state. So we sampled all the states and we estimated the free energy for one complete cycle from inward facing state to outward facing state is 5 kilocalorie. So we extracted the structures along this path, and then we have seen how the distance reduces at the bottom as well as at the top. So for example, I'm here showing the path 1, so so you can just notice how the distance reduces in the top and then how the distance increases at the bottom along the one cycle. And here I am showing the path 2nd, so where the distance at the bottom were like slightly high compared to the path 1, and finally it reaches the outward facing state. So once we have characterized these states, so now we can go into atomic details of each state. So this is the structural characteristics of OC state, and these are the various interactions that stabilizes the occluded state. So I'm not going into the details of these states. So I'm just comparing our predictions with the available transporter occluded state structures. So here I'm comparing the predictions with xylene is another xylose transporter from bacteria, and this is the obtained in partially occluded state. I'm comparing like, so the predictions of the top and the bottom matches more or less in agreement with the crystallographic predictions. And this is the another transporter which is multi-drug resistant transporter, where our predictions matches exactly with the crystallographic structures. We have done the same for the outward facing state, and these are the various interactions that stabilizes the outward facing state. And we compared our predictions with the another fucose transporter which transport fucose, and this is the bacterial structure, and we have seen like, our predictions matches exactly at the bottom and the top of the helices. So once we have these states, so now we are looking into the volume of the pore channel radius using the whole program. So here we have seen the highest state is the crystal structure. We have one starting point, and which is constructed at the top, and it has bulged volume at the bottom of this transporter, which shows that it says that it's open to the cell, and here it is closed at both the ends, and it has like more bulge at the center of the transporter, and it's OC state, and in OF state it's closed at the bottom and it's open to the cell. So now we are interested in what are the residues that cause these transitions. So I know this is very pretty picture, but it's very difficult to follow up. So I'm guiding, so we need to look into the two residues, which is R32 and then 360. So when we start from the initial structure, these are like very close and it interacts. When we move along the cycle, so this interaction breaks and they are far apart. And there is like another interactions which are far apart in the crystal structure when we move along the cycle. So it comes close and starts interacting and stabilize the OC state. So once we have this, we have looked into some literature data to support our predictions. So we have seen like D316. So when they mutate this residue, they kill the transport activity. And there are other two residues which I've mentioned in the earlier slides. They are like involved in stabilization of various conformations. And then there is histidine 1, so which is this one. So when they mutate this one, this also kills the activity. And there is another residue which is 68, and this is like responsible for increasing the potency of the substrate molecules. So here I will show the small movie. So how these conformational changes happens, and then also I will guide you like. So these are the two residues that comes close and interacts, and it forms occluded states. So where it is closed at both the bottom and the top of the transporter. And then this stabilization, this interaction stabilization weakens up the interaction at the extracellular pod, and it opens at the top. So in the literature, there are also like some experiments which are called DEAR experiments, which is usually used for membrane transporter proteins to measure the conformational dynamics of the proteins. So which is basically like, so in literature, so these are the distance measured. So basically they mutate the residues to cysteine, and then they create a disulfide bond, and then they add this molecule, which is nitroxide molecules, on both the sides, and then they allow to spin it along during the substrate transport, and then they measure the distance distributions. So the black line shows the distance distributions of the experimental data, and our green and the yellow, violet, and the blue line shows the distance distributions of various states from the MD simulations. In MD simulations, we measure the distance of C alpha atom, whereas in experiments, they measure the distance between the NO atom at the two sites. So more or less, our results agree with the overall distance distributions. So we have also looked into another transporter, which is called the plant sweet transporters. So which is the new family of transporters identified in plants. So plants get, so sugars are the primary source of energy. So plant gets sugars usually during photosynthesis. So the sweet transporter has been recently identified in plant OS sweet 2B transporter. So the topology has like 7 transmembrane helices, where helix 132 and 576 forms a pore, and then it transports the glucose molecule. And this is the typical structure of the sweet transporter. So we also performed the simulations in blue waters. So in this case, we started with one structure, and we got the entire landscape. And the total amount of simulation time is like 140 microseconds. And we also characterized various states like inward open, occluded, and the outward open. And the plot is obtained by measuring the gating residues at the top and the bottom of the transporter. And we estimated the free energy to be like approximately 5 to 6 kilocal for one complete cycle from IF to OC and OC to OF state. So we have also measured the pore radius of the different predicted states, where it shows clear distinction, which is like closed at the top, closed at both the sides, and then outward open state, it's closed at the bottom of the transporter. So we also compared our predictions with the close bacterial homologue of semi-sweet. So, and then the getting residues where matches exactly, and then here I have shown the pore radius where it is constructed at both the ends for sweet. And for OF, we have another bacterial structure homologue, and we compared our results where it is open to the cell and it is constructed at the bottom. So as a conclusion, so we started from one structure, and then we have predicted the other intermediate states for both PepTSO and then the OS sweet 2B transporters. And then blue waters, so using blue waters, we perform microseconds times long time scale simulations, which allows to understand the functional mechanism of these transporter proteins. And then we estimated the barrier. And then up to our knowledge, this is the first, we are done the first unbiased study using blue waters. Thank you for my group, and for the sweet transporter, so our collaborator Li Ching, she is doing experiments to validate our results, and thanks Blue Waters for completion. Thank you very much.